Hydrolysed Saccharomyces cerevisiae yeast prevented E. coli-induced increase in piglet intestinal tissue conductance in an Ussing chamber ex vivo model. Enterotoxigenic E. coli with F4 fimbriae can cause diarrhea and impaired performance in pigs by increasing intestinal permeability. The cell wall of yeast contains macromolecules that can bind to these fimbriae and may thus inhibit E. coli's ability to bind into intestinal mucosa. The aim of the study was to elucidate which components of yeast contribute to the integrity of gut epithelium. Enterotoxigenic E. coli increases the conductance of gut mucosa in Ussing chamber ex vivo model. Using this ex vivo model, our study compared two different yeast products, a yeast hydrolysate and a yeast cell wall product, in their ability to prevent the increase of intestinal permeability induced by E. coli. 16 pairs of Ussing chambers were each assigned to one of the four treatments: untreated control, negative control with fresh culture of enterotoxigenic E. coli, enterotoxigenic E. coli with yeast hydrolysate, or enterotoxigenic E. coli with yeast cell wall product. And the tissue conductance was continuously recorded for 2 hours. In the untreated control, the conductance remained unchanged throughout the 2 hours. And as predicted, the negative control group with enterotoxigenic E. coli showed steadily increasing conductance values. Hydrolysate yeast treatment showed unchanged conductance values throughout the 2 hours incubation. In contrast, yeast cell wall treatment showed steadily increasing conductance values. In conclusion, the hydrolysate yeast product was able to prevent the increase of intestinal permeability induced by enterotoxigenic E. coli. This is an interesting indication of protection against the negative effects of E. coli on gut tissue. The yeast cell wall product, however, did not prevent the increase in tissue conductance. Based on these observations, the molecules in yeast cell wall product alone were not sufficient to contribute to the gut epithelial integrity. Thank you for your attention.